Hi, this is Mitosis Notes Part 2. If you have not done Mitosis Notes Part 1, you should go back and start there. You're going to need the exact same materials we had for Part 1 notes. Go ahead and label your paper. And remember, if you need to go grab some materials, feel free to pause the video and then come back. So we're going to need to draw 3 cells again, but we're going to do something kind of funky with this third one. So don't get ahead of me at drawing circles. The first 2 are just going to be like the ones we drew last time. And again, you can always freeform them if you would like to. They don't have to be perfect. These are your notes. You are going to turn them in, but I'm not going to judge your drawing. Just do your best with what you have. Okay, so there's one animal cell. Leave a little space to label it underneath. Label the phase it's in. Here's 2. Okay, now this is where it's going to get a little weird. When you're drawing the last one, leave a little space open right here. We're going to show 2 animal cells splitting off from each other, so it's going to look a little funky. So I'm going to start my circle about right here, go around the left side, and not close my circle all the way. I'll show you what it looks like. Looks like that. Okay, then I'm going to take my glass, good old Lawrence High baseball glass, and slide it over and pick up where I left off. It's going to work pretty well. Look at that. Awesome. So that's what your third circle should look like. Scoot that up for a second. All right, it's fine if you need to freeform it. Okay, let's get going. So if you have your Mitosis Part 1 notes handy, that's really helpful. You want to make sure that you're using the same color that you are using for everything else on your last notes. So, for example, I was using blue for all the genetic material, and so that's where I'm going to start. I'm going to find my blue, and I ended with my, just to kind of remember where we were, I ended with metaphase where the sister chromatids were lined up in the middle of the cell. So that's what we're going to pick up today. Make sure I have this line for you. Okay, so I've got my blue. So remember, they had been attached to these spindle fibers, which I said, think of them a little bit like a fishing line and somebody's fishing and they toss out their rod and reel. And let's say they catch a fish and they start to reel it in. What do you think is going to happen to these sisters if there's reeling action coming from both sides? Well, maybe you've guessed that they're not going to stay together as sisters. So let's start with our centrioles. Our centrioles are still in the same spot. Okay, and then our sisters are now going to look like this. We had 5 pairs, and because of the spindle fibers being retracted and reeled back into the centriole, they ripped apart. It's very sad. They lost their sisters. And now, because they're not sister chromatids, we call them chromosomes again because they're just a single copy of the DNA. They're still tightly condensed, but we can't call them sisters anymore, so we call them chromosomes. So let's label what we have so far. Centrioles, chromosomes. Let's label this phase. I got ahead of myself. This is called anaphase. And the Latin name doesn't translate perfectly for kind of helping you remember what it means, but I always think Anna away because it starts with an A. So the sisters are moving away from each other, and they're moving away because they're being retracted by these spindle fibers that are being kind of reeled back in toward the centriole. have these little anchoring fibers, kind of help everything stay in place. And then the long spindle fibers that are just kind of reeling in, ripped them apart at the centromere. And now they're moving from the middle back toward the poles of the cell. So we label them chromosomes. We have the centrioles labeled. We're in good shape. And anaphase, by the way, technically means back. So I guess you can kind of think of them as moving back. But I feel like away helps me remember that a little bit better. All right, a few notes about anaphase. Okay, so the spindle fibers shorten. This pulls the sister chromatids apart. At the centromere. Remember, centromere is that center place on this. That was kind of holding the two sisters together. So they get kind of ripped apart, and now the name changes. Now they are called chromosomes, and they move to opposite poles. Remember, we called the opposite ends of the cell opposite poles. That's the anaphase. And we're ready to move on to the next phase. So I'm going to add a little color here. I like color. They make my notes pretty and they make me enjoy looking at them. So feel free to add more color if you would like. The next phase is called telophase. Some people say telophase, like telephone. Some people say telophase. Both are acceptable. Telo means distance. So telephone is like calling somebody who's at a distance from you. Telegraph, telegram. And we use this term because everything is moving to the opposite poles. It's moving away at a distance from each other. So the one thing that stays the same is our centrioles are in the same spot. They're still anchored down. They still have some of those anchoring fibers, kind of hold them in place. We have those long flexible proteins. However, remember the sister chromatids, now called chromosomes, have moved away, and they've started to kind of form together in one spot at each end of the cell, each pole of the cell. So I'm going to grab, a color I haven't used in a while. Check your work from notes part 1 if you have forgotten, but on my sheet, if you recall, I used this indigo color for the nuclear membrane, and that's the color I want to find next, because the nuclear membrane is reforming, but not just one, this time 2. And they're forming at the opposite poles. So make sure you use that same color. And now that the nuclear membrane is starting to form, these chromosomes can start to relax and uncoil and kind of be comfortable filling up that space, because now they're going to be tight and they're going to be protected on the outside by this nuclear membrane. And the nucleolus starts to reappear. So let's label everything. Centrioles, nuclear membrane. So now that our chromosomes have relaxed, we call this form of the genetic material chromatin once again. Kind of like we unrolled that ball of yarn into this space. And what else do we need to label? We need to label, do we get everything? Oh, nucleolus. All right. Add a little color to my telophase. And let's write some notes. Telophase So the nuclear membrane, not just one, but two, form around the chromosomes. They were still chromosomes in the early stage of the early part of telophase. At opposite poles, the nucleolus is visible again. The chromosomes begin to uncoil and relax, becoming chromatin. All right, and what else is missing here? So these little tiny fibers are still kind of anchoring centrioles there, but the long spindle fibers have broken down. All right. They can be reassembled later when they're needed. Cells are very conservative and recycle and repurpose and reuse a lot of their parts, which is pretty cool. All right, I'm going to scooch up a little bit to make sure you can read everything on the camera. Okay, good. All right, I didn't leave myself a whole lot of space here, so sorry about that, but this picture looks different, and it's called cytokinesis. Cytokinesis. And remember, we learned cyto means cell, and kinesis is moving. So the cell's literally moving because it's moving away. They're the two sisters, the daughter cells are moving apart from each other so that they can break off and become independent cells. I'm going to grab my nuclear membrane color, which was indigo for me, and look at that, my nuclear membrane is now fully formed. It's doing its job of protecting that genetic material, the chromatin. I've got two nuclear membranes. I'm going to grab my brown for my centrioles. You might still see a few of these anchoring fibers associated with it. I'm going to grab my light blue. My nucleolus is visible. My chromatin, I can't see anything really specific. It's all just kind of a solid mass if I looked at this with a good microscope and I stained a cell. So that's about what I could see. So nucleolus, this is our nuclear membrane. Of course, we have the chromatin. And then we have a new structure that pops up, and it starts to pinch in across these two cells. Kind of like, imagine if you took a rubber band and you put it around a water balloon. That's kind of what's going on here. This is a band of protein fibers. And eventually it'll just pinch that all the way off, and these will become two separate independent daughter cells. And if everything went correctly along the way, each step, we should have identical daughter cells. That's the goal of mitosis, is to create two identical daughter cells. Sometimes things don't go the way they should. There can be mistakes along the way. We'll be talking about some of those in upcoming units. So cytokinesis. So technically, if you remember our cell cycle, we've gone all through mitosis, interphase, prophase, metaphase, anaphase, telophase. Now we're in this little C pi wedge for cytokinesis, and interphase will start after that again. Okay, so cytokinesis. So everything that we did before in prophase, metaphase, anaphase, telophase was all about dividing the nucleus. Cytokinesis is about dividing everything else in the cell. So all of the organelles need to be divided up. All of the cytosol has to be divided up between the two new cells so that they are identical to each other. So the cytoplasm is divided, is what we say happens during cytokinesis. The cell is pinched in by a band of protein fibers. And if everything goes correctly, the end result should be 2 identical daughter cells. But sometimes there are things that go wrong or that don't copy over correctly. It happens. It's part of life. So over here, let's make a few notes just to wrap things up. I learned this when I was in 9th grade. Woohoo! IPMAT. That's a little acronym to remember the sequence of the stages of the cell cycle. So interphase, the in-between phase, prophase, when the nuclear membrane starts to break down, metaphase, when the sister chromatids line up in the middle, anaphase, when they start to move away from each other toward the opposite poles, and telophase, when they have moved away at such a distance that the nuclear membrane starts to reform. So IPMAT. And again, feel free to use color to make that fun for you if you like to. So here's a little sketch to show parent cell leads to 2 daughter cells. If everything goes correctly. And then I told you that these are animal cells. What if we have a plant cell? It's not squishy. It has a cell wall, so it can't squish in quite so neatly with this band of protein fibers. So let's just do a super fast sketch of a plant cell, and I'll show you how it's a little bit different. So remember, a plant cell is going to be shaped more like a rectangle because of that boxy cell wall made of cellulose. And so during this last stage of cytokinesis, it's going to have the 2 nuclei. It's going to have chromatin, but a band of protein fibers isn't going to do much for it. So instead, a new cell wall starts growing from the middle out, and it grows and grows and grows and grows until it connects and splits the sides, and it kind of compartmentalizes each new daughter cell. So this is called a cell plate, and that's one of the major differences when we're talking about cell division with a plant cell. All right, so that is part 2.